in water or in dilute alcohol. Normally, preference is for water, but in few cases, we may also go for alcohol along with water. You cannot use pure alcohol because alcohol is in a pure form a kind of a decolorizing agent. So normally we dissolve these dyes, and that too just 1 to 2% solution is made. That is, 1 gram of dye is dissolved in 100 ml of water or dilute alcohol. Normally, higher concentration is not used in staining. The thumb rule is you use the concentration which is slightly less and can increase the time rather than using a higher concentration of the stain and go for a less time. Because when you use a higher concentration of stain, and that too either in water or in alcohol, there is a possibility of evaporation. As a result, it may lead to the precipitation of the dye on the structures or in the cells, and they may lead to formation of artifacts observed under microscope. However, in certain conditions, the procedure requires to use a concentration which is at least 4 or 5 times higher than the normal concentration. For example, in a spore staining, spore is a very resistant structure and dyes easily, they do not easily penetrate inside the cells. So under those conditions, by a technique known as Bartholomew's and Mitwas method, concentration of malachite green is increased to 7.6%, and even the time period is also increased to around 10 to 20 minutes. So these are some of the exceptional conditions in which the concentration as well as time is both increased. Otherwise, normally 1 to 2% staining solution is prepared either in water and dilute alcohol, and the time period normally varies from 1 to 2 or 2.5 minutes at the maximum. Now, the dyes or stains dissolved in water or alcohol does not always suffice for the staining purposes. It means you may also need other chemicals during the process of staining, and these other chemicals are basically mordant, accentuators or fixatives and decolorizers. These things accompanied by the stains will lead to a proper or a good staining technique. Mordants are basically the complexes which are able to form some kinds of complex with the stain. This complex intensifies the color imparting property. As a result, either the cell or the cell structure is clearly observed. These complexes which are formed, they are formed inside the cells. As a result, these mordants, they basically take part in the staining reaction. Now, on the basis of the acidic or basic conditions, mordants are classified either as acidic because they react with the basic dyes, and basic mordants which reacts with the acidic dyes. Examples of basic mordants are alum, ferrous sulphate, cetyl, pyridinium, chloride, while acidic mordants includes picric acid and tannic acid. Now these mordants, how are they applied? As shown on the slides, they are applied as pre-mordanting, post-mordanting or in conjugation with the style. Pre-mordanting means they are applied before stain. For example, in Ringer's method for cell wall staining. Here, tannic acid or cetyl pyridinium chloride is being used before application of the stains. Post-mordanting means it is applied after the stain. Very commonly used procedure is gram staining. And third kind of application is in conjugation with the stains. For example, Lifson's method. The stain itself or the staining solution itself is prepared along with the use of mordants. Now, on the basis of whether we use mordants or not during the staining procedures, staining techniques are classified as indirect staining or direct staining. That is, if it is done with the help of a mordant, we call it as an indirect staining because we are taking the help of other chemical apart from the dye. While in those staining methods where the mordants are not used, we call them as a direct staining techniques. Okay, the second type of chemical that is being commonly used during staining is either the accentuators or also called as the intensifiers. Accentuators or intensifiers are basically those compounds, those agents which accentuates means it increases the intensity of the staining. However, they do not combine with the structure or with the dye. They do not form any complex with the dye. This is basically the difference between the mordant and these accentuators. Otherwise, basic aim for either mordant or the accentuators is the same. It intensifies the reaction and makes the reaction fast as well as very clear so that visualization is better. Now, these accentuators either can be a physical one, that is use of heat, or the chemicals. Examples in certain spore staining techniques, that is Dorner's method or Fleming's method, heat is used so that the spore pores opens up and the dyes can penetrate inside the spores. So it is a physical agent. Basically, it will not form any complex with the stain. Stain commonly used in Dorner's and in Fleming's method is ZnCF. This particular stain, when heat is used, this facilitates the penetration of the stain inside the spore. It does not form any kind of reactions with the stain. While in case of chemicals, examples of chemicals which are being used as accentuators, that includes certain acidic or basic compounds like oxalic acids, phenol, then potassium hydroxide or formalin. For example, phenol is one of the components of ZnCF stain. It is used for either acid fast staining or even spore staining. It accentuates, it helps the stain to make it more penetrative. Similarly, formalin. These are the compounds which acts as a chemical accentuators. Then the third kind of compound which is commonly used during staining is decolorizers. Decolorizers are basically the compounds or the agents which helps in removing the stain. Decolorizer, removal of stains, those agents which removes the stains either from the selective portion of the cell or in some cases from the overstained cells. Now these decolorizers can be either mordant, that is a mordant can also act as a decolorizer. For example, when we use excessive mordants, then mordant itself can act as a decolorizer. Then certain acids, alcohols or water, they can also act as a decolorizer, or certain oxidizing agents and even acidic dyes can also be used as a decolorizer. Now these decolorizers, they are used specifically during regressive staining. So on the basis of whether we use decolorizer or not, these all stains either can be classified as a progressive staining techniques or regressive staining techniques. Regressive staining means when we are using decolorizer, decolorize is only used when we have already used one stain, and that stain has either overstained the structure or the cell, and therefore we want to remove the stain from certain component, certain part of the cell. As a result, we will use decolorizers. So when we use decolorizers, the stains will be removed from those portions in which it is not firmly bound, and therefore that portion will be decolorized, while the portion in which the dye is firmly bound will remain colored. So this kind of procedure is a regressive staining as compared to progressive staining, where you use one stain, then it will be staining one particular part of the cell. Then you use a second stain, it will be able to stain the certain other part. This is a staining technique which we call it as a progressive. We increase the different kinds of stains progressively, step by step, one stain, second stain, third stain, and these all three stains, they will select a particular component of the cells. As a result, finally when the cell is observed, the cell may appear multicolored because of application of 2 or 3 colors, but that is a progressive way of doing it as compared to regressive, where you stain first of all everything and then keep on decolorizing so that the second dye can be used at a later stage. The another way of staining, apart from normal staining technique, is the staining by impregnation method. So far we have seen that we use the dyes or the stains, that is those compounds having chromophore group and oxochrome group. But in certain cases, we have been using the colorless solutions of metallic salts. Basically, they are colorless because certain metallic solutions, when they are dissolved, then they do not form any color. For example, ammonical silver nitrate, which is commonly used in spirochete staining in Fontana's method. Now, when these colorless solutions of metallic salts are used and either heat or some other chemical compound is added, it reacts, the reaction is carried out and a precipitation, opaque precipitation is formed. This opaque precipitation can be of a different color. Basically, the staining solution was not made from a chromophoric agent, that is, it was neither a dye or any other thing. It was a metallic solution, some salt, and it was colorless. So chromophore group is not present. But because of reaction with some heat or oxidation or other things, these salts have precipitated out from the solution, and these salts will be deposited on the cellular surfaces. As a result, what happens? Because they are opaque in color, it will make the cell or the component colored, and another important advantage is it increases the size, apparent thickness of the organism is increased. As a result, it will also aid, it will also help in visualization of the cell. So that is the technique which we call it as a staining by impregnation. Now, for any staining techniques, basically fixation is a very important part of staining. Before we begin the staining procedures, it is necessary to fix the slides. Why? Because to prevent the washing of the smear. Smear is made on a slide, and when it is fixed with a fixative. Either that fixative can be a heat or it can be some chemical compound. Normally in a routine procedures we use heat as a fixative. That will immobilize the cell, that will adhere the cells, organisms on the slide. And therefore when the stain is being washed or the decolorization treatment is given, these structures, they adhere on the slide, they are immobilized on the side. And basic mechanism is they are denatured, the cells or the proteins present in the cells, they are denatured or they are coagulated. As a result, the fixation occurs. As I have told you, it can be either a physical agent like heat or certain chemicals like gaseous acetic acid, ethanol, chloroform, formalin, and even certain metallic salts like mercury chloride are being used as a fixatives. Now, advantages of fixations are also many. That is, it kills the organisms and naturally it imparts the biosafety. Then, since the cells are killed by using a fixation, by the fixatives, either heat or chemicals, because the cells are dead, they lose their permeability. Otherwise, you know that cells membrane is selectively permeable. They will not permit dyes to enter into the cells. So when these cells are dead, they will help, their permeability increases. Then another thing is, because the proteins and other things they coagulate, it will lead to unfolding of proteins, which increases the reactive groups, and thus it also in turn increases the affinity towards the dyes. Apart from these, there are other advantages like it avoids shrinkage or swelling of the cells, it prevents autolysis, avoids the formation of artifacts, and since it makes the cells rigid in certain tissues, the sectioning becomes easy. So these are the various advantages of fixation. Now let us come to one more thing, that is, what is the supravital and intravital staining? If the unfixed living cells, they are killed due to the application of the dyes, then such kind of staining is known as a supravital staining. It means the dyes proves to be toxic to the cells. That is a supravital. In contrast to that, if the cells or the organisms are stained without killing them, because of the non-toxic nature of the dyes, it is an